continue. Here, the lecture, seminar from the last week. So last week, we studied about the EKG. Right? How EKG is working in terms of ion channels and the conduction of the electrical signals. So most puzzling element in this board EKG signaling, we found there's a hyperpolarized motivated ion channel and cation channels, which is an HCN channel. Right. So remember this rhythmicity of the nodal cell, because actually highly contributed by the HCN channels, right? Normally, I mean, more places, more ion channels activated by depolarization. In other words, higher voltage. Right? How is specific channels, it's called HCN channels, activated by the hyperpolarization, step depolarization. That's the question like that in the last lecture. And then, okay, so this one, once this HCN channel, high channel open, right? It's non-select cation channel, so it allows the introduction of this sodium, so it elevates the rest potential of these neurons. And then what type of channel relates after activation of the HCN channel? Next play. Over. Channel. Channel. It's calcium channel, right? There are two types of calcium channel. One kind of calcium channel. One kind I will be using. The P-type calcium channel, right? Friendly calcium channel.
Yeah.
They slightly elevate, and once you reach the rest member potential, like the -45, then what type of calcium channel activates them? So A-type, right? Long-rest calcium channel. Okay. So once the long-rest calcium channel opens, it's kind of overshoot, and then potential channel opens, then it's and then HCN channel open again. It elevates. So it's with this flow relay of the ion channels, it continuously or perpetually it's activated, right? So even without the external stimulation, this nodal cell can generate its own rhythmicity, right? That's we studied last week. So
Does it mean that it's a mechanism for pacemaking?
yeah,
Cells?
yes,
Yeah.
sure. So today we are studying about the molecular mechanism of the HCN channels more specifically, and then I briefly mentioned how it's related to pains, especially neuropathic pains. So so what are HCN channels? So again, hyperpolarization activates cyclic hyperpolarization channels on neurons and heart cell. So not just the neuron, not just the heart cell, but also neurons. In other words, brain too. So this is the diverse expression patterns of HCN channel. You know, it's a kind of problematic in developing the drugs because it's not specific. So if you want to develop drugs, which is like for example, like a inhibitor of the HCN channel to reduce the pain sensation, but this same blocker also blocks not just the peripheral, neurons, but also brains and heart cells. So there's another word there, actually, heart and brain side effects. Okay? So this we can discuss a little bit about it later. So anyway, so there are like 4 types known. HCN type 1 to 4. So some types that some channels consist of a homomer, or the tetramers consist of HCN1, or some type of channels consist of tetramers, like a mixture of the different types. So anyway, so here, simply, it's consists of 6 transmembrane domain and like the other types of general multi-gate channels. And then why it's important? Because HCN channels, it's also involved in epilepsy, chronic pains, heart rhythm disorders, all these kind of symptoms actually severe, can be severe disease. Then there are slight difference between type 1 and 2 for them.
Question in the previous slide. So in chronic pain, HCN channel expression may make kind of persistent signaling. Is that how, like pacemaker?
It gives a persistence, not necessarily the pacemaker, but hypersensitivity. It's overstressed. That's why it's for even for a long time, it's the target of the neurospace pain. So already many actually chemicals has been developed. But as I said, this channel is not only interested in peripheral, but the central region too, especially heart. So it was to develop the drugs specific target the peripheral side. But luckily, this is what I should tell at the end. But
yeah. Luckily or unfortunately or unfortunately.
For the professor's team, I will go and
yes. Yeah. To the conclusion first.
conclusion. In 2014, in England, the anesthetic doctor developed particular chemicals to specifically block the HCN channels. He used the computer modeling, but so now he owns already a company. Yes. Yes. That is the company. Oh yeah.
Which is
Peter was a developer of this chemical. Then he made the commercial version for the neuroplastic pains. He will establish a company, Akeros, 2018, I guess. So yeah, something like that. But now it's actually a good time to develop this kind of energetics, because we now take advantage of AI, which means to find the specific like the targeting of the molecule structure. So maybe many actual companies already working for that. Just keep in mind. This is an example how things working.
It's only 5 years old company then.
5 years, yeah, it's new, yeah, it's new. I don't think it's listed in Costa, that's not yet. But, they got investment.
I wonder what phase of the clinical trial.
It's not yet critical phase.
Just a pre-clinical trial.
It's just, it's still undergo further phase research.
Oh, 70 million.
So looks like at the moment, this chemical or this drug, potential drug, is the most promising candidate for the HCN drug as for the neural pain analgesics. So back to the remember before next time. So once compared to the major HCN activity, if the HCN activity is increased, this is the frequency of fire will be increased. It results in the hyperexcitation of the unit. Okay? The overexpression of the HCN channels or the or the drugs activations. Oh,
I have general question. For example, in case of brain,
yes.
there is a natural resistant to brain, that is blood-brain barrier.
Yeah.
But either something exists in case of heart.
Oh, yeah, that's actually it's a good question. Yeah. To develop drugs which that can pass BB is difficult, right? So it's easier
For heart.
for develop drugs which not pass the BB, right? Yeah, right. So that's the thing. But we don't know. I mean, I'm not a chemist, So far, I mean that's the problem. We just think about it. But there was already, drugs. Ivabranium is famous drugs, for to reduce the depolarization of the cardiac cells. Okay, like reduce the heartbeat. It's already on the market. The lots of research is chemical. So, in brain, it's same. If the exchange channel is hyper excited, the overall the brain oscillation, will be increased. There are lots of experience spots in many places, even retina. So each case is related to different types of diseases. Pain, in case of pain, also it's related to the neuropathy, hypertension. So, yeah, just keep in mind as a background.
What's the difference between HCN1 and 2?
This, yeah. So, for example, like HCN2 is particularly expressed in hippocampus and brainstem or deeper brains. It doesn't mean HCN1 is not expressed in the brain, but the 2 is specifically in those regions. So it's more like let's say in HCN1 is hard, but in 2 is like the neural activities, the cognitive function, because it relates to the expressed in hippocampus. Like that. But a little bit, there are slight differences in the functions where you can read. Yeah, and the general structure of HCN is a little bit complicated. So we have this channel in transmembrane region. Then the work cytoplasmic domains. Okay? where the cyclonucleotide, the cyclo-AMP can be bound. Okay? So it's a bit bulky. So it's kind of complicated. That's why it's this channel structure is relatively cloned later compared to the other simpler channel, sequence channel. So that's why the paper came out in 2017 in their paper. The original like protein channel paper published in early 20,000. Okay, so almost like more than 10 years later, it's published, which means the structure is bulky and complicated. So you might also have a little bit difficulty to understand this paper if you try to understand. So the basic structure like three components. Here, voltage sensor. This like any other types of voltage channels. And then it's a HCN domain. And then C domain and cylinder and CMP, the different one inside domain. So we talk about the four region first, especially S4. If you take a look, in this transmission domain, you can see, especially S4, there's a difference S1 from the other transmission domain. What's the difference?
Lengthy,
What?
more lengthy.
Lengthy, yeah, exactly. It's long, right? It's long. The other channel, the other transmission branch is inside the transmission membrane, but the S4, it kind of protruded, especially into cellular regions, okay? Central domains. See, this is the key, the length, as it said.
I have a question just, I didn't read the paper fully.
Okay.
Did they, like, verify the structure with X-ray crystallography?
Oh, the clear.
Here?
Yeah.
Uh,
Yeah. But because it's a little bit bulky, that's why I don't know, maybe it might be different to make crystallize.
Yeah.
Yeah.
So that was my second question, because looking at the structure is a little bit bigger for the crystallography, so how
Exactly,
they
yeah.
do that?
Yeah. Good point. That's why it took maybe a longer time.
Yeah.
Maybe they might, I didn't ask them, they might try it first with X-ray. When it failed, somebody switched the method, I guess. So if you see, if you take a look at the paper, the two papers, the same, it's from same lab, the same, basically same channel. You can see there is a little bit difference actually in terms of the diagram. See? Especially in S4. See? When I first read this paper, in 2017, it wasn't clear. It wasn't clear. It didn't make any sense. It didn't really make me clear understanding, clear understanding. So that it's not, I realized that after 2 years later, it's not just my fault. They kind of modified their model, or in other words, they kind of their projection was not clear, or was in other words wrong. They're wrong. So,
Not retracting their paper.
yeah.
Another cell paper.
Yeah, but this is just diagram, so so after two years later, they gave clear, kind of clear picture of the C4 and S4's movement. Now I could understand clearly how opening and closing is operating through the S4. So anyway, so previously the length it's okay, it's same, the length. they pretty well, even in 2017. But the problem is, they didn't find the cooking mechanism. The type. They thought previously when before, I have a form, we just kind of pull it down, and pulling the S5, then open. But actually, they realized they found none. There's some they found bent. Cytoplasmic size. Okay, this is the key. But in 2017 paper, they did not just not only talked about the what's the protein sensor, but also talked about the cytoplasmic domain too, how the cyclic AMP binding can open, help to open the gate. So more likely this is more general pictures for mechanism of the channel's operation. But this paper is more like specialized for the S4 gating needed by S4. Okay. So I combined these two papers.
But still, to me, it's still not very clear that, so S4 now has a like bending that direction, and S5 is also bended towards more inwards. And I'm not sure why that will open the channel.
We talk with the data.
Okay.
So the mechanical hyperpolarization was very positive, as you know. Right? So where this kind of where and how is moved by the polarization came from? It's from the actually S4 helix formation. So to discuss about HCN, I want to revisit the previous presentation. If you remember, for potential, particle potential. In particle potential, there are repeated conservative amino acid in 4 regions, GYG, glycine, tyrosine, glycine. And this 4 regions provide the carbonate actually for the 4. So it just like, divides some rooms and 4 passages for the potential ions can penetrate. Okay? So this passage the environment where the potassium ions can feel the each region actually exactly same when it's surrounded by water, the ions. So there's no.
So the red
Not-
dots are water
The
molecules.
red dot oxygen.
Mhm.
So eight oxygen or 8 water molecules circumvent a single potassium ions, right? So this environment is exactly the same inside the 4. So in terms for this potassium ions, it doesn't feel any any energy hindrance when it enters into this 4. So zero, almost 0. So it can move very smoothly. That's another, see, the 3D structure. So this the 3D structure surrounded by water, exactly the same inside the pore. Okay. So this is the potassium channel 4 regions. And this one is HCN channel 4 region. So what's the difference? What's the difference between these and this? It's the structure. What kind of what's the structural difference between potential channel 4, yeah, HCN channel 4?
Color wise, the second one looks more blue.
Well, no, I mean the passing of the passes.
It's two shades, the top.
The second
It's like
one,
a narrow.
the second one is in,
Narrow and like, and it's become wider.
Yeah, exactly, yeah. It's apparent, right? It's like open, like zipper and zipper. It's like half open zipper, right?
The last layer protein is also different, right?
That's what?
There one, the acid is T base and there is C base.
Oh. This one?
Here is the last one is the C. On the A, that one is the T.
Ah, turning this thing?
Yeah.
Yeah, I mean, that's different, yeah, that's it's for sure, but more like for internally, this is side chain difference, right? Yeah, you mentioned it before, side chains. This is the comparison of the side chains between potential channels and exchange channels 4. What's the difference? Same tyrosine, right? So this tyrosine, this one, is more like rotating, like order like this. Tyrosine here, it's like rotating almost 180 degrees. So, and then this more structure, let's see, this rotation pulls the widening in this thing. Chamber 1 and 2. Okay. Then the question is, this is another example of the sodium potassium pump. This pump, the similar is only 3 and 4. More than 2 is wide open. So, what does it mean? And why this what kind of what the such a difference affect the ionic selectivity? In other words, why this potassium channel 4 only allows selectively potent ion to pass through. Okay? It's not just because this molecular environment, but also the combinations of the feeling, the probabilities inside these chambers, which means these 4 chambers within the potassium 4, it's always like alternatively it's filled with potential ions. Okay, 1 and 3 or 2 to 4 configurations. Okay, so if just in case potassium, sodium, which is smaller than potassium, right? From the molecular side, when enter, this is blocked. It's blocked not just by the potential ion in front, but also the other chambers. The two barriers actually for the potential ion, sodium ions has to be overcome 2 times. Okay, that's why there's this kind of configuration also gives the selectivity for the potential ions. Okay, but compared to that, let's check. Since it's only there are 2 chambers, either if just case either one, like one or let's say okay, chamber 3 is occupied by potential ions, and this one without the ion, potential ions. Then if once the potential ions pass through, then there's no other second barrier, no other second potential ions to block the sodium ions to overcome. The sodium ion can come easily. So that's why this kind of like open spur, I can say, the configuration allows gives the non-selectivity of the ions. What's that? So this the same phenomenon, the same structure configuration can be found in sodium potential pump too. Also, there are three chambers. So that's why we provide the non-selectivity, which means that both sodium and potassium channels pass through easily. Okay. Another, okay, this is clear. By the way, have you ever, anybody try to see the 3D structure with this figure? No? It's the stereoscopic image. Amazing.
Oh, cool.
The book,
If
with
you-
that,
If you print or screen, if you just watch and put two images together,
Yeah.
then it becomes the 3D images. You never tried?
the 3D glass, like the red and the
Exactly.
green one, yeah. You know,
Right.
okay.
Then once you try, you can see the 3D structure like VR. I could see
Can you do it now?
it's maybe too wide for you to see. It's really, but if you print, A4, even in total screen, you can see the 3D structure. Otherwise, it's hard to see. What's the
Difference.
in the 2D, but you lose the information. Anyway, so this is to help your understanding the three structure, and then this the pore region from the extracellular point of view perspective. So it's more like a wider compared to the same challenge, the pore wider. So this also yes, in the 3D structure. You can see the direction coming out. So this S4, 5, 6 first member domain, which are aligned and parallel to each other. And we can see the kind of interlock almost right like a zipper. It gives the stability. In other words, it helps the form the closure structure. So that's why the S4 helix movement is the key signal transmitter. And also S4 and S5 linker also plays a role between S4 and linker, S4 and S5. How it works? Here. H4 and G5. Actually, this one is S4 and S5. This link.
They are not down to this block, and therefore the chemical is fine later.
I mean, the linker means the two transmitted domains are linked. It should be linked like this. The linker. This is called linker. Okay. This linker. This linker gives you also stabilization. Also, if you were able to see the 3D image, it might be really easy to understand. The point is, okay. The point is, these linkers and that's linker. Exactly cytoplasmic domain. This one, okay? This is cylinder. And then this is the CD, before the CMB, the cylinder. What they want to say is, what the point is, cylinder kind of sort of like a kind of plane structure, like a flag, okay? Little bit flat, no flat, okay? Plane, okay? This is the point. It's what they want to say. And then this is the point is this cylinder is also what's that? This S4 and S5 linker stabilize this cylinder region because within close proximity. Okay, this is the point. And also you can see here is the what's that HCN domain. HCN domain. It's like here. It's like a perhaps structurally in like wedge. Okay, like a wedge. So in this structure, it has the closure condition. Right. So then now we can talk about the cyclic nucleotide binding domains. So if compared to the bound and unbound, this is also okay. This is the okay, more and stickiness. So before bounding, and it's blue before bounding, and then it's the yellow, at the binding with the cyclic aim. It's more like a kind of, let's say, internally the arms, like an internal round, like this, hold in order, or stabilize like this. So if you see within 3D image, like more clear. The structure involved structurally, actually the same this blue is different binding, yellow is different binding. It does not dramatically change the form, but it has little bit. So that means cyclic AMP alone cannot open the channels, but it facilitates the opening of the channels in hyperpolarization. You see? Then, okay, so here, in people, why is most important? What's the main difference between the dipole and hyperpole? And how in depolarization, as for helix, like has longitudinal shape, because the yellow is arginine, which is positive charge. Okay. So many positive charges align, in this transmembrane. So in depolarization, which is the intercellular region, the polarized, positive charge, you push outward, okay, bounce back. Understand? Okay. Then what happens in hyperpolarization? Then in case of hyperpolarization, this band, this region is thin, which is weight, which is a little bit less bulky. There's a cutting happens and this band region. So in this weak linker, there's band like a angle. Oh. So this case almost like a can almost throw it's moved downward intercellular region. And then this length, this one, this like from I6, it becomes almost like a parallel to the membranes. See? Okay, so this is the whole story, actually, the more details, but this is the overall pictures of this ion channels. This is structure, actually structure paper. You better understand intuitively, like look at the 3D structure and understand the property of the structures. That's most important. So anyway, through this structural mechanism, the HCN channels is able to be activated by hyperpolarization. So I already talked about the potential therapeutic implications at the beginning. So this can be a good candidate for the energetics for neuropsychiatrists. So any other questions?
Question. So, if HCN overexpression is a problem for a neuropathic pain,
Yeah, there are evidence that
uh,
pain.
then typically
Overexpressions.
do we use
Yes.
antibody against or like are there any small molecule which will block the channel?
There are actually many candidates. There are many, for the development. Again, it is said, there are, side, they cannot overcome the side effects to the brain and heart, which can't develop specific drugs. And more, the direct drug development, the specific, the most important. Maybe we can get ahead with the AI technology, targeted drug modifications. Maybe we can work with the
We may not want to like give HCN blocker because it may stop the heart. Side effect to block the heart.
Yeah.
So that might be a
Which are really better developed drugs, specifically overcome, bypass, blocking the HCN channel expressing right? Only in temporal regions. That's why it's difficult.
So, I have like two questions, like, is it possible to do this, like, in the HCMES settings that can we separate out the cardiomyocytes and can we look for these signals on the AEM?
I think so. Yeah,
It's possible.
yeah. Once there was already
Isn't it possible to find these records on the HCMES and do this, the control that one we discussed, like in the PNL matters, positive control of the emitter and the negative control?
Yeah, but we need your candidate.
Oh,
We don't have a candidate.
yeah,
If we develop, this can be a platform.
yeah.
Okay, so yeah. And this is it, and I also want to talk about the our future. Put the okay, and then